Herbert.
Yeah.
Okay. Next, we have Professor James Glazier from Indiana University, and he'll discuss multicell agent-based modeling, and specifically through the use of their software simulation environment, CompuCell 3D.
Okay, thank you very much. I'll put a link to the software in the chat. This is going to be a much lighter talk than the one TJ just gave. And so I thought it was the end of a long week, so I wanted things to be pretty easy to digest. So it's going to be mostly pictures and not a lot of detail. I'm going to be talking a little bit about multicell modeling. You've been talking a lot about modeling networks, and I'm going to be focusing a little bit on what you could do by combining network modeling with modeling how networks interact between cells. And it's going to be a lot of examples. I'm not going to be showing any code or almost no code today. As the title slide just showed, these are going to be reviewing very quickly projects that have been done by people over the years, enormous number of collaborators. Ultimately, I think many of us are here because we want to be able to use modeling in a medical context. Ultimately, if we're going to be able to treat a disease better, we need to be able to predict what's going to happen to health states. A lot of the network models are currently being used, for example, in physiology models are used in that way. The kinds of modeling I'm going to be talking about today aren't really integrated in those kind of workflows yet. We hope maybe eventually that they will be. But we have to be able to describe how organisms work, how they're built, how they maintain themselves, how they fail. And there are different kinds of failure in organisms. They can fail through what are called developmental diseases, where the mechanisms that normally are used to build and maintain the organism are misdeployed. The classic developmental disease being cancer, although there are plenty of others. Infectious disease, TJ talked about that a little bit. This kind of modeling could be used in tissue engineering, and something that may not be so familiar to people that we've done a lot of work on is in toxicology. How do chemical perturbants affect tissues? A drug treatment is in a sense the flip side of toxicology. In both cases, you're doing a perturbation. In one case, the outcome you hope is what you want. In the other case, it's something you don't want. In all of these cases, you have networks inside of the cells that are controlling their behaviors. And you have a lot of complex interplays between cells, both network-like signaling and also biophysics. And so these are all situations in which you need to understand both network dynamics and mechanics and physics. You might call this multicellular systems biology. How do you build, starting with individual cells, all of the many structural and functional phenotypes that you see in tissues? The number of things that cells do is not so many. They could grow, divide, differentiate, die, metabolize to create energy and chemical products. They can move. They could interact with each other. They could stick to each other. They can pull at each other. They could eat each other. They can signal to each other. They could secrete chemicals into the environment, consume nutrients in the environment. They can cause flows if they have cilia, for example. There also are physical constraints on what's possible and how fast signals can move. And ultimately, that gives rise to complex phenomena. One of the classic studies, again, of this kind of work is in cancer, which is why we have that example. But ultimately, what we'd like to be able to do is to understand these multi-scale systems and their organization, and eventually to be able to engineer them and control them. So we need to do this at multiple scales because we have typically molecular control. For example, if we're doing medicine with drugs, we're typically having drugs that act at a molecular scale inside of individual cells. If we're doing measurements in the clinic, typically we're measuring serum levels or whole body levels of things, highest scale. But really what we want to be able to control is what's happening at the tissue level. And if you're an engineer and you go back to beginning engineering, you'll be told that you should always measure and control at the same scale at which you care about the outcome. If you measure and control at different scales, you get all sorts of instabilities. And so there's a fundamental problem in biomedicine that the scale at which we can do control is smaller than the scale we care about. The scale we can measure is coarser than the scale we care about. And the hope is that if we do multi-scale computing, we can interpolate among these three scales and use computation to be able to infer things that we would not be able to do directly from experimental measurement. The classic example of this would be to think about a car. If I give you a list of all parts in the car and say from the parts list, tell me what the car is like to drive, you're not going to be able to predict it. Fundamentally, the drivability of the car is an emergent process of the ensemble of parts. And you actually don't need to know a lot of detail about the screws that are holding the seat to the floor of the car to be able to understand how well the car drives. And so there's also a lot of detail you can ignore at the molecular level to understand the control of systems. And so we have to remember that biology has this very elaborate set of scales of integration that you have to think about. So what we would call virtual tissue models are essentially middle-out approach. You start out by modeling individual cells as agents. In the body, cells really are the main active agent. Everything that happens in the body is a result of the behaviors of individual cells. There are a lot of other components in your body that are critical to its function, but things like force generation are all done pretty much by cells, movement by cells. You are going to have a concept of control, which will be networks that describe metabolism sometimes, gene regulation, signaling inside of the cell. So that would tie back to the course that you've just finished. You might also put those tissues, the simulated tissues, inside the context of transport and metabolism at the whole body level, what are called PBPK models, which would also be network models. So in a sense, you have network models at small scales and large scales, and we're going to make spatially explicit models in the middle. And typically, we will have 3 or 4 scales. We're going to have a network control scale at the lowest level. That's going to control what cells do. Then those cells are going to find themselves interacting with each other in an environment. And this is a simulation we'll come back to where we're simulating the development of a chicken embryo and the formation of the periodic pattern that's going to give rise to the ribs and the spinal cord, called semitogenesis. Here you see chemical oscillations inside of the individual cells. You see long-range gradients of chemicals. You see coupling between the cells, and you also see changes of cells. There's cell division going on, there's cell differentiation going on, and cell migration due to different cell behaviors. These are agent-based models. They're mechanistic. They're intrinsically multi-scale. And our focus is on emergent behaviors and structures due to those interactions. And again, our key is to try to combine the biophysics of what cells are doing with the control from networks. Our outputs classically are movies, and the issues that TJ just brought up about how do you compare stochastic simulations or serious ones in these kinds of contexts. I don't have any universal solutions to that. It depends pretty much on the simulation you're working. If we're going to represent cells, there are many different ways to do it. You could represent them as cellular automata, single particles. You can represent them in a way what are called center models, which are essentially molecular dynamic simulations. TJ has a beautiful package called Tissue Forge, which is actually this set of movies that are running in the middle here of this image. You could do mechanics, finite element-like simulations. There's called vertex model simulations. We use a lattice-based method, which we find as a bit of an intermediate in terms of the level of spatial detail and complexity that we like to work with. As TJ mentioned, for the past 20 years now, we've distributed an open source software package called CompuCell 3D. It's a modeling environment that lets you build and execute this kind of simulation. If I had an hour and a half instead of half an hour, I would have you download and run that code with me. If you're interested, you can pull it from the link that I put in the chat. I'm certainly happy to answer questions about that. Basically, what we're trying to do here is provide key concepts that are going to be used to model biological organization, generalize cells, because you can also represent components of cells like cell nuclei, leading edges, secretory apparatus, mitochondria, and what those components have as properties, volume, surface area, polarity, and so on, what they do and how they interact, chemical fields outside of cells typically, but also inside of cells, chemical concentration, for example, links, which represent solid-like material properties, and then networks. And since you've been dealing with networks, I'm going to talk primarily about how you couple this kind of modeling with networks. Those networks, again, could be representing something like a signaling network or gene regulatory network, biochemical reaction network, or a pharmacokinetic transport network as well. So those are all built-in concepts inside of CompuCell. You can specify your networks. Since people have been doing networks, you could specify your networks with SBML, CellML. I don't know if I'm allowed to say CellML in this environment. Antimony, if you like Boolean networks, you can use MapBoss. And you can run those models either inside of individual cells, so you'd have many replicates of the same model interacting between cells, or as freestanding models to represent the global information system. This is an old simulation going back to the Tyson 2001 cell cycle oscillator model, where we've got a cell here, simulated spatial cell, that has simulated cyclins inside of it. Those cyclins are controlling the cell growth and division. It's rather an old simulation because the coloring is a little bit mislabeled, but gives you an idea of how to do that. You can actually pull in pre-existing SBML models, couple them from a file, for example. Here they're being actually read from the biomodels database, and run them inside of your simulated cells. If you like antimony, and I happen to always teach with antimony because I find it simple to learn, and you can really learn how to use antimony in half an hour. Not many programming languages or model specification languages that are as elegant and simple as antimony. Here's a specification of delta notch signaling, which is a very important juxtacrine contact-based signaling mechanism in developmental and homeostatic biology. In the case of juxtacrine signaling, you can't really understand how delta notch works if you only look at a single cell, because really you have to understand cell 1 has a delta signal that is sent to cell 2 through the notch receptor. Cell 2 has a delta signal that's sent to cell 1 through the notch receptor. They have a cross-inhibitory feedback, and so you can only really understand what this pattern does in a spatial context. And what it does is produce something that's a little bit like a checkerboard. It's actually not quite a checkerboard. A high delta cell will be surrounded by all low delta cells. So it's more like if you imagine a checkerboard or a tic-tac-toe board where only 1 in 9 cells is turned on delta, it looks like that. So it's not an even number of cells of the two types, but you can run that kind of model. In CompuCell, actually, we do that a lot. That's a very common model to use because this kind of cross-inhibitory feedback is very common in biology. This is what that simulation looks like. If you run it, you start with the random initial conditions, and eventually some cells go high delta, some cells go high notch. There's competition, and you'll get eventually a relatively stable, depending on the geometry, you'll get a relatively stable pattern of high delta and low delta cells. I said I wouldn't show any code. There's some code hiding here. And I could come back and walk you through what that looks like. But it's pretty easy to specify models of this kind. TJ wrote code to couple this kind of modeling to Boolean network models, in this case implemented in MABOS. This is actually the same delta notch model that you just saw, coupled to that same cell cycle model that you just saw, but implemented in Boolean networks. And here are cells growing and differentiating based on the cell cycle model and the juxtacrine inhibition model together. So that's basically what this kind of modeling framework provides. People use it. This is some recent papers from various groups using this kind of model. And if we look at this, people have looked at sort of self-organized cysteines in general. This is another juxtacrine signaling model. Germinal center response is immune maturation, how your body controls immune cells in the lymph nodes. Adenocarcinoma, so a study of cancer. Epiblast for developmental toxicity. So this is a rather detailed model of early embryonic development, trying to understand how chemicals can cause birth defects. Ovarian cancer model, so another cancer model. Simulating 3D cell shape. That's actually a model of tissue engineering. Modeling of tissue invasion, epithelial monolayers, another cancer model. And yet another cancer model. So people use this in a variety of contexts. Let me just show some examples. That's really, again, this is the end of the day. You've had a long week. Movies, I think, are relaxing, I hope. This is this developmental model. Here we're modeling, again, chicken embryogenesis. The initial embryo is elongating. What's going to form the spinal cord and the rib cage is this midline that's dark in the chicken embryo. These little black spots that you see forming in the embryo are called somites. Those are going to reorganize to form the spinal cord and the rib cage. And what we want to understand is how the tissue that's initially coherent elongates and differentiates to make that pattern. Inside each cell, there's a relatively elaborate chemical oscillator network implemented using gene regulatory network and signaling network. There's a gene regulatory network controlling cell differentiation. And then outside of that, the cells move, stick to each other, and divide and send and receive signals from the environment. You can do a little bit more detailed modeling where you actually think about the individual chemical components inside of the cells and how they move around. This is the hair patterning in fruit fly embryos. In this case, each embryo, each cell in the wing has a little hair on it. It's not really the same as our hairs. It's a spike on the cells because we're at a scale that's much smaller than an individual cell. Those spikes are oriented, and there are a variety of chemical species that associate with the cell membrane that determine that orientation. This is a simulation of the movement of those molecules within the cell along the cell membrane to give rise to that polarization. So this is taking a network model and decomposing it into molecular level spatial detail. We've worked a lot on modeling retinal vascular diseases, and other vascular problems. In the case of what's called wet age-related macular degeneration, sometimes the blood vessels that are initially behind the retina, supplying it with nutrient and oxygen, invade the retina. If you've ever had a retinal scan, there are blood vessels on the front surface of your retina, which is what you see when you do a retinal scan. But there also are very dense blood vessels behind the eye, behind the retina. And those, if they get into the retina, if they invade the retina, they can cause blindness through what's called choroidal neovascularization. So a neovascularization, new blood vessels. And we were interested in why that happens and the way it does. And so this was a relatively elaborate simulation, mainly of biomechanics, where we've simulated the what's called Brooks membrane behind the retina, the blood vessels, which are here purple, underneath the retina. The green cells are the black lining of the eye, the pigmented retinal cells. And the gray cells here are the photoreceptors. In the 3D movie, we've made the photoreceptors transparent so you can see what's going on. And here you see in red blood vessels invading from behind the retina into the retina, proliferating because there's a lot of growth factor, and then causing the detachment of the retina and then breakup of the retina. If that happened to you, you would lose your vision in that region. If it happens extensively, you'd go blind. And simulation like this, TJ was talking about doing statistics on stochastic simulations. Every time you run this simulation, you're going to get different results. In this case, we have done many replicates of simulations with different parameters, and we've tried to understand the probability of particular patterns of failure of this architecture as a function of different perturbations. So normal here is in this top right corner. In the normal retina, the blood vessels never invade. And now we're going to perturb particular aspects of the retina to understand why invasion happens and where it happens. If we imagine aging, normal aging, in that case, you have accumulation of lipids, essentially soap, on the top surface of the membrane that the pigmented retinal cells attach to. That means they don't stick to that membrane as strongly. And if you reduce the adhesion between the pigmented cells and the basement membrane, you have a high probability of this kind of invasion. If, on the other hand, you had trauma to the head, in an accident, for example, that tends to reduce adhesion between the retinal pigmented cells and the photoreceptors. You get a different pattern of pathology. And if you got an eye infection, that reduces adhesion between the pigmented cells themselves, and that leads to yet another type of pathology. And so you can actually use a simulation like this to understand quite a few different kinds of eye disease associated with different kinds of pathologies. And so that was work that we did about 10 years ago now. It's actually validated in experiment afterwards, and potentially creates targets for new drug discovery and treatment processes. We've also worked a lot on diabetic retinopathy, which affects mainly the blood vessels in the front of the eye, trying to design optimized therapies for that. Again, the point of going through all of these examples is just they're all using the same software package. And so it's trying to show you the variety of things you can do using these methodologies. Another thing that we've worked on quite a bit over the years is a disease called polycystic kidney disease. Again, trying to discover drug therapies for this. In this disease, the kidneys keep growing. They don't stop growing when you become an adult, and they form cysts. And eventually, if you don't get a kidney transplant, you die with your kidneys the size of watermelons. They stop working. Rather unusually, people may think of cancer as a genetic disease. It's not really a genetic disease. In this case, if you have a mutation in one of these two genes, polycystin 1 and 2, you're almost certainly going to get the disease. If you don't have it, your probability of getting the disease is less than 1 in a million. But it wasn't understood how these mutations gave rise to the cystic phenotype. Our clinical collaborator and experimental collaborator had noticed that some of these cells expressed a cell adhesion molecule that's normally only present in children. That was a hint. And so we were able then to build a simulation of just a small fraction of the kidney, a tubule in the kidney. We were able to introduce a simulated cell with this wrong cell adhesion molecule in it. Cell adhesion molecules do two things. They help cells stick together, but they also send signals, what are called contact inhibition signals, that help the cells measure the density of the tissue. And the role of that is that if there's injury to the tissue, there's a signal that says, I need to proliferate and fill in the gaps in the tissue. And so this is what's called a homeostatic developmental mechanism. It's a way that the tissue can repair itself without any detailed information. Basically, tissue is always saying, is the density right in this place? If the density is right, the cells stop dividing, they persist. Density is too high, the cells may die to reduce the density a little bit. If the density is too low, it'll send a signal to say, proliferate until I fill the cells in. So it's basically building a self-repair mechanism. If you have that wrong signal, you get proliferation of cells when they shouldn't. Tissue is complete, but the signal is not correct for that geometry, and that leads to cysts formation. And you can then compare that to experiments, in vitro experiments, and show that you match. You can say there are different ways this signaling could fail. I'm not showing all the networks and things here at the moment, but you can show that there are only particular ways that it fails that would match what's seen in an experiment. And then you can do more classic bioinformatics of the signaling pathways to identify drug targets. In this case, we were able to identify some potential drugs to use to treat the disease. Computational toxicology is nice because we really do have to do a lot of network modeling. This is a model of a small patch of your liver, blood flow in your liver. We have acetaminophen, Tylenol, in the blood. Acetaminophen by itself is not terribly toxic, but some of the cells in your liver convert it to a very toxic molecule that can cause individual liver cells to fail, and at high doses can cause your liver to fail. And actually, thousands of people die every year in the United States from overdosing of acetaminophen, from Tylenol overdosing. And we were working with the Environmental Protection Agency and then with the NIH trying to understand the mechanisms by which that toxicity happened. So here we have an explicit model of blood flow in the middle. We have a model of transport of acetaminophen and its metabolites in the whole body as a network model. Inside of each of the simulated cells, we have a metabolic model, and we're calculating how much of the toxic byproducts are produced. If the toxic byproducts are too high, the cells die. And you can then model that. You can look at patient variability between individuals to try to understand susceptibility to toxicity there. That work was done a while ago. More recently, there's been a group at Michigan State University that's built very detailed models of both cell signaling and metabolism in the liver using CompuCell to do what's called zonation. Different subregions of the liver metabolize different chemicals in different ways with rather complicated structure, and they have a beautiful metabolic model, gene regulatory model, and transport model of the way the liver works. The EPA has used this kind of modeling to try to understand developmental toxicity in particular. Nicole Kleinsteuer, who's now head of NICEDM, NIH, during her postdoctoral period, 10 years ago or more, worked on vascular disruptors, chemicals that could interfere with blood vessel development in embryos. Here's a model by Max Lung modeling the effect of estrogen mimics on disrupting urogenital development. There was a lot of concern about feminization of male embryos by estrogen mimics in the environment. Actually able to simulate dysfunction of the genital development in embryos. So again, here we have rather detailed outcome networks inside of the individual cells, but in the context of the whole tissue as it develops. Shane Hudson at Vanderbilt did a lot of work on the fusion of the palate in the roof of the mouth. Some chemicals in the environment can cause cleft palate in embryos. Again, you have quite a detailed signaling and gene regulatory model inside each cell, plus a very detailed biomechanical model of how the tissue grows and rearranges. We've worked a fair amount on modeling infectious diseases. Here you have an initial cell infected with a virus. The virus replicates using a network model inside the cells. It's then released and spreads in the environment, affecting other cells. That also leads to the release of cytokines that pull in immune cells. This was a very simplified model that we built a long time ago, but you could use it to study, for example, the efficacy and variability of drug treatments, in this case, antiviral treatments, between individual replicates. And I think some of the ideas that TJ was talking about in the previous talk really came about from our grappling with the extreme variability of the outcomes for the same parameter sets here. Fundamentally, perhaps the main result's not that surprising. If you have a lot of variability, you need to use a higher dose of the medication than you might expect to get the outcome you want. That's fine in general, but if the medication is toxic, you can't do that. And so you'd like to understand the causes of that variability. TJ has also worked a lot on how you can, in a reproducible way, take ODE reaction kinetic models and turn them into spatial models. This is beautiful work that he did, again, using CompuCell, taking quite an elaborate model of immune response in rodents to influenza, which had been validated, or calibrated maybe is a better word than validated, quite extensively against animal studies. So you actually have the time series for the different main chemical species, the different cytokines, also the different cell types, and then turn that into a spatial model where the individual cells are represented explicitly. And so you have the various immune cell types represented explicitly, many of the chemical signals as chemical fields, and we're able then to calibrate this very complex spatial model of immune response to reproduce the time series that are observed in the animals. These kinds of models are complicated. Biology has lots of components. Reproducibility between components is not great. Two cells that are both nominally the same type, two neurons, two hepatocytes, two endothelial cells are quite different from each other. Things are not standardized as engineered systems are. There are lots of individual components. You have lots of cells and tissues. We can't model all the cells in a tissue. Maybe in a cubic millimeter, we can model all the cells. There are a lot of parameters in the models, and we can't always measure those in experiments. And so there are a lot of difficulties building these kinds of complex models. But I hope that you saw at least some of the different applications you can use and maybe got a sense of how you combine control at a network level inside of cells and between cells with the way these simulations play out in space. Is it worth going from a ODE or network model to a spatial model? Well, do you need spatial information? Is there a lot of spatial heterogeneity in your problem? Do cells move around? So there's tissue reorganization, structural change in your problem. Is there long-range transport in your problem? If the answer for those things is yes, if you have movement and spatial heterogeneity, then really to understand your problem, very often you are going to need to go jump in and do agent-based modeling. Pure network models may not be sufficient. How many cells do you need? If you need 100 cells or 100,000 cells, with CompuCell, you're all right. With TJ's tissue forge, you can go to 1 million or 2 million cells. But if you need 10 to the 7th cells, 10 to the 8th cells, you're pretty well stuck. You can't represent each one of them individually. You need to know a lot to build these models. You at least need hypotheses about regulation, about signaling, about interactions, at least ballparks for that. I do think feedback is critical, especially feedback between scales. If feedback is critical between scales, you can't see the whole world inside a single cell, then you may need to do these kinds of methods. And there's some things that are just hard. If you really need the extracellular matrix structure, that becomes a hard problem. I would love to see, I've been showing this slide now for 15 years, the ability to take, say, a microscope image of individual cells. This is an old experimental movie from 2008 where they're imaging every cell in a zebrafish embryo, in a living embryo as it develops. I wish we could build a simulation where we could pull that in, create a simulation and simulate the growth of the embryo in parallel with the experiment. One of the postdocs in my lab has been working with me and TJ, trying to do the first steps of that, Sharon, but we're still a long way from doing that. Maybe one of the people in this group, in this workshop, will solve that problem and have it happen for us. If you're interested in this, we'd love to help you out, work with you, whether it's in cancer, vascular biology, toxicity. If any of you are interested in software development, it's an open source project. There's a lot to do. TJ and I work on this a lot together. So you'd be happy to talk about it. There's a working group that we've had now since 2020, talking about not just this particular software package and methodology, but multicell modeling more generally, which you'd be welcome to join as well. People want to help us out on the model development, we'd be delighted. It happens, I'm sure people are tired, but Sunday we start our workshop on multicell modeling. If anybody really wants to sign up late and start Monday morning, you'd be welcome. We can reopen you're welcome to participate. I don't know if anybody's actually planning on doing that, but if you are, you'd be welcome. If you want to learn about it, but not do it next week, the courses are all online on YouTube. So if you Google CompuCell 3D Summer Workshop YouTube, you'll pull up the course materials, which are all available online for self-study. If you want to learn a little bit more about multi-scale modeling in general, we have an archive of about 150 seminars using CompuCell, Physicell, Morpheus, and other packages that implement these kinds of ideas. You can always reach me, jaglazier@gmail.com, if you want to talk about these things. And thank our support from over the years for all these projects. And I have to say that I have some patents on some of these ideas. And so potentially there's some conflicts of interest that have to be disclosed, although they're not really very related to the things I was talking about today. I hope that gave you a little bit of a light flavor for the kinds of things you can do with this kind of modeling. As I say, if we had a little more time, I'd actually have you run some simulations of this kind. It's pretty easy to learn how to do the simple simulations and couple them. But I hope it gives you a flavor. And if there are any questions, I'd be happy to answer.
Does anyone else have any questions? And thanks a lot, James. Oh, yes, Stephen.
Hello, thank you very much for your talk. I was wondering how flexible it was to incorporate different like you were saying that it currently works for antimony and then sell them all. Are there other ways to get a new software that could potentially run into if it could develop concrete models or develop different models to apply into CompuCell 3D or apply into a way to translate it into antimony readily? That's what I was a little bit
I'm sorry,
interested.
could you say this? The beginning of what you said was cut off, so I heard just the end of that question. I apologize, but could you repeat the question?
Yes. Can you hear me now?
Now it's fine, yes.
Cool. So thank you very much for what you had said. I was interested in potentially using a software for my group to apply the models that are currently running because we also do parmest or parameter estimation and a design of experiments. Applying those models into CompuCell 3D or finding a way to put them into Tellurium to then put into CompuCell 3D. Is that something that could potentially be analyzed for next week? I'm going to be in the workshop next week as well. So
Yes,
that's something that
especially now, well, I'm still learning it. TJ has and Maciek have implemented a Python. CompuCell used to be more or less a monolithic platform that ran. You could do it as a batch job, but it was really designed to run on your desktop as a freestanding software package. They've implemented it now, so it's basically a Python library that can be called from Python. And so anything if you can call, if you have a software tool, for example, parameter estimation, it can call CompuCell and have CompuCell return results for doing things like optimization. So yes, parameter identification and optimization are things we're very interested in. We do face the issue that TJ was bringing up, which there are two things really that we face. One is that these are stochastic simulations. So every time you run the simulation, you're going to get different results. And so you have to run multiple replicates, and you have to decide what parameter, what metrics you're going to quantify, and how you're going to then define whether the distributions of those results are the same or not. If you have a scalar metric, then the methods that TJ has talked about are fine. Something that comes up a lot, though, for example, if you're simulating blood vessel development, is that you could get two different vascular patterns, and you want to know, are they the same? And they're never voxel-wise identical. You can't use a Hamming distance or another simple voxel-based distance metric. On the other hand, when you look at them, you can pretty well tell the textures are different. And so we've been working, TJ and I have been working with a grad student actually in University of Virginia, who's developing AI classifiers to try to be able to look at some of these spatial patterns and ask the question, what is the similarity or difference of these spatial patterns? If I have two blood vessel networks, can I tell were they grown with the same parameter set or not? So that's most of what we're talking about here doesn't use AI, but in terms of pattern matching, that may be a place where AI could be very helpful. So I'm glad you're taking the course. Somebody put in the chat, by the way, that they tried to register and weren't able to do it. If you email me and you really want to take the course next week, you're welcome to do it. We never turn people, well, I won't say never. We almost never turn people away. In that summer course that we're doing next week, we've had everything from high school students to emeritus professors take the course, everything from pure mathematicians to surgeons take the course. We can't say it anymore because he graduated from high school, but for the 3 years, we had actually a high school student help present the course because he did some really beautiful computer simulations of immune response using CompuCell, which were published as a journal article. And so he came and he taught his methods to the class. So he actually had a high school student as an instructor in the class for a while. Now he's a college student, so it doesn't count as a high school student. But we're happy to talk to you about this, and we're going to have that hackathon the following weekend. And so it would be a great opportunity to try to couple the methods that you're interested in with CompuCell. So that would be great. Sorry that was a long answer, but there was a lot in that question to unpack.
Yes, thank you very much for your answer.
Okay.
At the same time. Okay, well, we've come to the end of this week. I hope you all learned something and enjoyed parts of it, if not all of it. We might send out a survey. It'll be anonymous. If you have any suggestions for topics that you'd like to see, and also whether there's any interest in having much smaller ones throughout the year that focus on specific areas. So anyway, thanks very much for attending.
And I'll put it in the chat again. If anybody wanted to take the course next week and couldn't register, email me this afternoon and I'll get you on the list so you can take the course next week. No pressure, but if you want to, please do that, because I saw a couple of people said they couldn't register. I don't want you to be turned away. Sorry, Herbert. I didn't
That's
want
fine. That's it. We've come to the end,
no,
I think.
no.
I think we're done. Is any other commentary? Otherwise, we'll let you go and enjoy the weekend.
Yeah. I'll be monitoring the Slack for a while, so feel free to ask questions there for follow-up things. And I think Dr. Hollowstein will probably be in touch with some people about if we try to get a hackathon report in somewhere. So make sure everyone gets credit for that.
And if you think Herbert and I should do some more, Herbert and I used to teach this as a joint course. And then it got to be too big, so we split it into two separate courses. But if somebody would like to see us come back together and try to do some courses specifically on the integration of network modeling with multicell modeling, I'm sure you could twist our arms to do something like that. We're thinking about teaching a course in the fall using a Boolean network modeling software called Cell Collective. Specifically focusing on modeling immune cell development and response. So if there are specific topics that you're interested in, seeing some kind of integrated course like that with, let us know.
Yeah, okay. Okay, lots of great thank yous. Yeah, we appreciate that. Thanks a lot.
Special thanks to everyone who asked a question during the class.
Yeah,
great.
no, yeah, the icebreakers in particular got everything moving. They're particularly important. Yeah. Okay. I'm going to grab lunch. So we'll send out a survey at some point next week or two, so to get more info. Okay.